we need to get back on the career trajectory here,
Yeah.
right? You said you were the move from Editas to Vor. So that was Vor and Tome, I think, were the next two stops on your journey. Tell us a little bit about why you made those switches.
Yeah. And so it's interesting. I've definitely had some mentors along the way who've advised me to branch out or to generalize. I kind of, when I took the job at Editas, I honestly didn't even understand what CRISPR gene editing was. It
Hmm.
was, a technology in its infancy. I had a couple of postdoc friends who were using it for screening, but I had no idea that it was going to become sort of a next frontier in genetic medicines. Um, and as I looked, at beyond Editas, I again, these mentors were saying, okay, you've had, an incredibly successful stint at, one of the pioneering editing companies. Do something else to, go into a different area of technology or a different area of disease. But for me, when I looked at the field, there is so many good science. There's so many other good things, particularly around the Boston area, that you could go for. But I do
Yeah.
not see any other path to alleviating or even curing rare genetic disease that's more promising than gene editing in its many forms.
Mhm.
And so I've stayed put. That's definitely been a theme for me, staying at companies that are exploring with different generations of gene editing systems.
Yeah.
Um, and and in many ways, it kind of feels like that, is the perfect intersection of life experiences for me. That, I'm trained as a scientist, and fundamentally, gene editing is not an easy or a simple process. It is, a very intricate system, complex biology. And in my job as in business development, I really never know who I'm going to have to explain or interpret to. I could be dealing with a Nobel Prize winning scientist who wants all the details, or I could be dealing with somebody who has more of a finance background and needs it to be, explained in a simpler manner that
Mhm.
they'll be able to deal with. And so, you've got that part of it. And then on the deal negotiation side, one of the things that's been really interesting then is getting to see as this, field of gene editing has started blossoming over the last decade or so, seeing what it takes to have success or not in the field of biotech startups, seeing, that combination of good people and good ideas and good strategy and how some of those things pan out, like what decisions you make with the data you have in hand at the time versus what decisions, you might have made if you'd had 2020 hindsight.
Mhm.
And then finally, that patient perspective, right? It is, I remember the clinical indication selection exercise at Editas, and it's very patient driven, it's very patient focused, but that patient is also other. That patient is sort of, cut and dry facts that are on a piece of paper. What does the disease progression look like? What are the symptoms? What's the underlying genetic mutation? Can our technology fix that mutation? And once you get a diagnosis, the psychology of that exercise is so very different because it becomes, that could be my daughter,
Yeah.
right? How are
Right.
we approaching this safely? Is this, can we, with the way that clinical trials need to be set up, are we going to be able to intervene early enough in that child's life to have meaningful clinical benefit? It just, it changes the equation. And so I think, one of
The,
the things, go
the
ahead.
equation and a sense of like does it up your sense of urgency?
Definitely ups the sense of urgency
Yeah.
of impact. And I've seen folks out there on LinkedIn and X, talk about the explosion of gene editing companies, and do we really need that many? But I would argue yes.
Yeah.
There can't be too many companies, right? There are so many rare diseases and so many patients out there who are looking for answers that I really don't think there can be. And it also then has changed in certain ways the way I view the business of medicine, right? It's phenomenal to see, like the Casgevy, the first approved medicine, CRISPR medicine for sickle cell that CRISPR Therapeutics and Vertex, brought to the clinic.
Hmm.
Seeing folks get cured and no longer having these horrible vaso-occlusive episodes with, pain and blocked blood vessels, that is amazing to see. And those drugs are priced accordingly. I mean, hundreds of millions of dollars of research go into, every one successful drug has 99 failures sitting in the background.
Mhm.
And yet, when you look around the world, the patient population of folks with rare disease is not limited to, rich people in developed countries that have great insurance. It
Yeah.
spans the gamut. Nobody's spared, right? And so you start thinking about, we are very early in the arc of a new technology. Things are usually most expensive at the beginning because they're so new. And you see, as things become more common and commoditized, you do see prices come down. But it really did start get me thinking about like, how do we Jennifer Doudna and her Innovative Genome Institute, right? This idea of democratization. How do
Mhm.
you take such a powerful technology that has applications that go way beyond human therapeutics? When you think about sustainable crops, climate change, and how do you responsibly use it for some of these things? How do you use it for the betterment of society, and how do you do it in a way that's approachable for as many patients as possible?
You're doing a beautiful job. God, you're a great storyteller. Fantastic storyteller.
Thanks.
Uh, and you're doing a great job setting up my segue to the next career pivot. Um, I talk to a lot of physician scientists turned biotech founders, and they tell me, well, I was administering the thing and the things, and then I made the decision to get into biotech or biopharma because I wanted to make the thing, right, that I was administering. So here you are, you're working for companies that are making the things, a few of them in a row, and doing well, I'll add. Great business development career with these companies, doing what a lot of people aspire to do, making the thing, developing the thing. And then last year, early this year, you decide now I want to go do something else. And it's not with a company who's making a thing per se in the context of, in terms of a therapeutic. Now, like I said, I think you were sort of leading into that question when you were talking about these bigger problems, these bigger challenges that your current company, Profluent, is attempting to play a key piece in the puzzle, right, of solving. So tell us about that. What prompted and how was like what was going on in your mind? What did you struggle with when you said, you know what, like I think I'm going to move away from working in BD for therapeutic development companies in the gene editing space, and I'm gonna jump on this. I won't
AI.
say, yeah, AI, yeah, and I was gonna
bandwagon.
yeah, I don't want to call it a band. I mean, yeah, I'm fully on board that bandwagon, and I don't even want to call it nascent. Like it's here, right? It's happening. So, but it's still big transition for you, big decision, right? Walk away from therapeutics development and take on a risky role with a startup company that doesn't really have a product per se when you joined. Like, what was going on there?
Yes. And so, I mean, as you said, I was drawn to the previous companies, to Editas, to Vor and Tome, because they were all, pioneering incredible editing technologies and turning those into medicines for rare disease, for immune disorders, for cancer. Um, and as I experienced both those specific company builds, but also sort of getting the bird's eye view of the developing gene editing field as a whole, I one of the things that I was struck by is a pattern of needing a new approach to protein engineering. So why would we need a new approach? What wasn't working or what isn't working? So in many cases, CRISPR being one of them, the starting point for a therapeutic is a protein sequence that has essentially been copied and pasted from nature. And often it's by random chance or discovery. It's
Mm.
not that somebody's trying to find that particular thing in nature. It's that somebody's working in bacteria and then discovers this thing that does something, and they start thinking, huh, how could that be applied? Could that be applied to human medicine? Um, Now, the tricky thing is that a protein that evolved in nature to carry out a certain function might not be the perfectly designed solution for a given human use case that you're looking for. And so if we want to adjust the function, how do we go about doing that? We need to tweak it slightly. How do we go about doing that? And so there are sort of, different approaches or different eras of protein engineering. It's not a new thing. It's been going on for decades, right? So
Mm-hmm.
one is sort of the manual route where you could carry out random mutagenesis or directed evolution, and trying to guide the protein towards those criteria you're looking for. This is incredibly time and resource intensive. It often is sort of, again, random mutagenesis does not really imply that you have much of a hypothesis. It's your sort of like randomly tinkering with different areas of the protein to see if you then get results that you want. Um, and often what you end up getting is incremental improvements at best. More often you break the protein because you tinker with a fundamental area of it that's necessary for function. And so, then maybe moving on to the next era or sort of a different approach, we've got folks who do metagenomic mining. What does that mean? Well, they look at, I lifted this particular sequence from this particular organism. It's almost right, but it doesn't do these other things we want it to. So I just need to broaden my search. I need to look at more organisms. And in the diversity of organisms, eventually I'll find something that does have my list of criteria.
Mm-hmm.
And certainly some folks have had success, looking into, volcanoes and ice flows for more genetic diversity. But I would still argue that that is a fairly needle in a haystack approach. And it often results in functional trade-offs. And so what do I mean by that? So when you look at natural evolutionary processes, often the selection pressure that's being put that leads to sequence changes is focused on one particular attribute of that protein, of that enzyme. So for example, perhaps the host organism is small. So the enzyme itself needs to be smaller. It just isn't going to fit. It's not going to be able to do its job. But in the process of evolving and achieving that smaller size, it's possible, I'd argue even probable, that other aspects of that protein's function are going to be altered. And so change is kind of like a double-edged sword in this scenario. It's, you achieve one goal, but you sacrifice other features that weren't critical for fixing the current problem in that original organism or
Yeah.
survival of that organism, but they are key for the therapeutic problem that you want to solve. And so, in comes Profluent and generative AI, which I'll be frank, I knew nothing about. I'm a huge sci-fi aficionado, but I knew nothing about real life AI before being introduced to Profluent. And, you know, one of my first thoughts is this is the next era of protein design, where we're moving away from sort of accidental discovery or sort of, tinkering around in a manual way. And we're really moving to intentional, bespoke design aided by artificial intelligence. And, they have these large language models. And those large language models, they're not limited by natural sequences. They don't involve manual labor. There's no functional trade-off. It's when you look at the combinatorial space of a single protein, it's greater than the number of atoms in the world. And so there's no way that even a team of the best scientists can sit down and explore all the possible sequences, right? But generative AI can do that in a matter of minutes,
Mm-hmm.
right? It helps you narrow the funnel of what could be possible, places evolution hasn't had a chance to explore, and pull the gems out. And so rather than sort of starting with a molecule and then trying to kind of like jam it into a therapeutic use case that it may not be quite perfect for, but it's sort of good enough, we're really trying to kind of flip that paradigm for drug development upside down. Start with the problem. What is the problem you're trying to fix? And then if you had to imagine the perfect drug that would fix exactly that problem, what characteristics would it have?
Mm-hmm.
And once you have that list of criteria that you think define the ideal system, then we task the AI with building exactly that thing. And that just blew my mind that could that even work?
Yeah.
Right.
I'm going to bounce around here with my questions because I'm going to go with like there's primacy and recency. Ask questions about something you said earlier. I'm going to go with recency.
Okay.
This description that you just illustrated, start with the problem and then ask what are the attributes of the solution to that problem. and then go out there and use the data. It seems to me in that situation, the data, at least at this point in time, might be the weak link.
Or the strong link, depending
Or
on.
where, yeah. So let's talk about that. Like I said, I'll probably bounce around here for a minute, because I'm already in over my head. But hey, here we are.
Here we are.
Where does the data come from? Like, where does the data come from? And like in that scenario, um, do we have, do you think enough data to make impact, like to query, to apply these models and make impact?
Yes. So I will caveat by saying that I am not too unlike you. I am definitely in over my head. And anything I know about AI, I have learned from my team members, but especially from our
My
investor.
my listeners are calling your bluff right now. They've been hearing from you for the past 45 minutes, and they're like, this woman is not anywhere near over her head.
I'm a, I'm trying to, it's definitely sort of like a fire hose, right? At the beginning of a new job, picking stuff up. And we're lucky enough, we have a board member named Fraser Kelton, who was the former head of product at OpenAI for ChatGPT and DALL-E. And then Profluent's own CEO and co-founder, Ali Madani, who was one of the leading researchers at Salesforce that did all of this foundational work on applying large language models for protein generation. And so, they've definitely helped bring things down to my level in terms of understanding, what are large language models and how did it go from an area? If you look back 5, 10 years ago, your iPhone model, I don't know, 2, 3, couldn't finish a sentence for you. Like its predictions, right? Were often hilarious. I would sometimes just like take the prediction because it was so hilariously wrong, right?
Yeah.
So how did we get from that world to a point where we have something like ChatGPT, where you can say, write me an essay in Shakespearean style on soccer, and it needs to be 2 pages long, and it can just do that, right? That it seems like an incredible advancement. How did we get there? And as they describe it, the two key ingredients, one really is fundamentally advancements in AI. There's something called the transformer algorithm that apparently had a profound effect on models' ability to learn and generate useful results. But the second key ingredient is what you are alluding to. It's data.
Mhm.
It's the scale of data. It's the quality of the data. It's the bias or lack of bias in the data. And so, when you think of, how things like ChatGPT were able to have success, there are millions, if not billions, of examples of text floating around on the internet in, books, all these different things, right? And so that's a rich abundance for the model to scrape and learn from and learn, just by like when you think of a young child learning language, right? Some of it's intentional teaching, but a lot of it's just passive, absorption. They hear things, they learn grammar unconsciously, and that's what these models do. And so now if we turn to the world of protein discovery, that same abundance is mirrored in nature. We have, the world has provided us with this abundance of natural protein sequences over the course of evolution.
Yeah.
And much of
It,
that
it,
data is just like readily available. It's just out there, it's public.
it's I guess that's part of my question though. Is it analog? Like, is that data that's out there in nature? Like, if I think the publishing industry certainly was an early to the internet, early to the cloud industry. Um, I haven't been around the life sciences space long enough to know that they were jumping on that bandwagon from the outset and saying all the data, all of it, it needs to go. It needs
Well,
to be digitized and it needs to become accessible. So I think that's kind of central to my question.
yeah. So I would say that there are ways in which some data doesn't exist. And we can get into that, I think, later, maybe when we talk about sort of like negative data. When
Yeah.
it comes to things that are natural sequences, as opposed to, a drug that somebody's trying to develop, when we think about what exists in natural sequences, yes, there is so much data out there that's digitized. Our current frontier model is being trained on over 80 billion protein sequences
Mm.
that are just out there. I can't say that they're often uh, perfectly curated or ready to go. So there's a lot of intentional mining, tidying, curation that needs to go when you pull down this information from all of these different sources, both public and proprietary, so that it is apples to apples and your model can actually learn from it. But there's so much of that that exists out there. And that's what we call unsupervised data, right? This is just sort of like you want to expose your model to as many examples as possible so we can start playing little childlike games over and over trillions of times. It's think of it like fill in the blank exercises, right? Where, if I say the girl kicked,
Hmm.
you have learned that probably something's going to come after that. It could be the ball, it could be her brother, right? But there's a narrower universe of things that could come in that fill in the blank than if you just started a sentence with the. And
Yeah.
so, in the same way with these protein sequences, the model, as it's exposed to it, it's learning the language, it's learning the syntax and grammar, it's learning what protein domains are responsible for what functions, it's seeing evolutionary dead ends where things didn't work. And ultimately, what it comes out with is the ability to write things from scratch. So I'll be very clear here. Our models are not starting with a existing sequence and then performing the AI version of targeted mutagenesis. They are being fed input where you say, I have this therapeutic problem. To
Hmm.
solve it, I need an enzyme with these qualities. And it says, okay, let's start with amino acid one. If I know what I want the end protein to be capable of doing, what should the next amino acid be? What should the next amino acid be? And it generates millions of these possibilities and then ranks them based on its own prediction of how confident it is that that's going to be a successful hit. And that's where the second type of data comes into play. So you have this unsupervised natural data that you're exposing the model to train it, right?
Hmm.
Now you have supervised lab data. So there's this sort of like feed forward loop where if our models have generated certain sequences and the model thinks that these are going to be, CRISPR analogs, for example, right? Now you have to go test them, right? So you take the top five and maybe a couple sprinkled around that it doesn't think are going to be as good, and you put them into the wet lab and you run functional assays and you see how they perform in real life. And now you've gone from like the theoretical, the hypothetical, to like rubber meets the road. Like, what does this actually look like in practice? And when you get those results back, that's supervised data, it's labeled data. So you feed that data, positive or negative, back into the model. And it learns which of its things were successful, which ones weren't. And maybe it sees patterns in terms of why, what made the successes a success, and vice versa, right? And so, it then learns from that data. And I think that's a really interesting point that ties into sort of, the idea around both is there enough data and what kinds of data are there? I think, in the field of biotech and medicine, there is an emphasis on successful data. That's what
Hmm.
gets you papers. That's what gets you drugs, right?
Yeah.
Publish or perish. And yet in the field of tech, there is a very different attitude towards quote unquote failure. It's, move fast, break things, learn from them, right?
Hmm.
And that view of when something fails, you can learn from it. The models will learn something. So it's not a waste of time. It is, in fact, if your models were only exposed to positive data, that
Yeah.
would not be a good thing. They would not be as well rounded. And so that for me was a very interesting psychological shift that you're almost, as intrigued by negative results as you are by positive results.